Hello and welcome to Beyond Biotech, podcast number 22, going out on November the 11th, which is Remembrance Day in many countries. I'm Jim Cornell from the Biotech, and I got back from my trip to Copenhagen a couple of days ago, so still recovering from that a bit, but it was a good trip and it's always nice to be in Copenhagen, although it was a mixed bag weather-wise. But I was really there for the Medicon Valley Alliance annual summit, so a few videos and interviews to come from that over the coming weeks, to add to the pile of interviews to get to. I think I need 36-hour days. It's a bit of a different podcast this week because we only have 2 interviews. And the reason for that isn't because I don't have any others done, it's because they are both quite long, but they are both very interesting, so I left them a little bit longer. So there are just the 2 conversations this week with Neil Ward, VP of PacBio EMEA, and Marc Giulianotti, Senior Manager in Space Biomanufacturing at Sierra Space. And that's not forgetting, of course, our weekly chat with global commercial real estate services company JLL, and this week it's with Travis McCready. So now it's time for the news you may have missed over at Labiotech.eu. The first patient dose has been completed for a bladder cancer diagnostic drug. Ascentage Pharma announced positive news on its chronic hepatitis B drug, and proteins identified by Funcell and developed by Forcefield can preserve heart function. Sensorion has been granted rare pediatric disease designation by the FDA for its lead therapy gene. AI could tackle pharmaceutical manufacturing bottlenecks. A new hormone discovery could predict the long-term health of men, and Telix Pharmaceuticals announced positive results from a kidney cancer imaging study. BC Platforms received $20 million in financing. An AI program resulted in multiple novel RSV drug targets, and MedInCell's partner AIC registered a trial for a drug for total knee replacement pain. We're definitely seeing a lot more AI articles recently, and we had one on some of the big guys, as Google and Meta, formerly Facebook, said their AI could boost cancer research and drug discovery. We had a story on how COVID vaccination may boost cancer treatment. We looked at the 5 hottest biotech startup companies in Brazil, and some new funding for NRG therapeutics will be used to work on the mitochondrial link to Parkinson's and ALS. A new radiopharmaceutical shrinks prostate cancer tumours. The study of copper may open new cancer drug treatment doors, and a novel branched lipid boasts mRNA delivery to cells. Ascentage Pharma has received Chinese approval for a solid tumour study. Novo Nordisk is supporting the plant-to-food platform with €27 million in funding, and researchers have developed a new nanoparticle to deliver drugs direct to macrophages. Avstera received funding for its cancer programs. Rescue Biotech is using bacteria to discover Alzheimer's treatments, and a promising drug candidate has been discovered for the prevention of type 1 diabetes. A Sanofi and GSK COVID-19 booster vaccine has been approved by the European Commission, and you can read all of these and a whole lot more at Labiotech.eu. So let's get to this week's guests, and first this week we are talking about DNA sequencing. PacBio has launched its Onzo and Revio sequencing systems, and I won't even begin to try and describe them. I'll leave that to the expert, and that is Neil Ward, VP of PacBio EMEA.
I mean, the obvious question before we start talking about the things that you're working on is if you could give me a bit of background on PacBio.
Sure, yeah. I mean, PacBio, it's a company that's gone through something of a rejuvenation in the last couple of years. So the company itself was formed back in 2000. Our founders had a great idea of how to take DNA and essentially stick it in a teeny tiny hole and measure a polymerase, so the enzyme that copies DNA, in real time, kind of videoing a movie of that polymerase integrating a complementary strand of DNA, so additional letters, A's, T's, C's, and G's, to build up a complementary strand of DNA and measure the fluorescence that was attached to those letters as the polymerase goes around there. that was the concept. I think that concept had huge promise as a sequencing technology, but like most technologies, it takes some time to come to commercial fruition. So while it was founded in 2000, the company had gone through various iterations of products and form factors, waiting for the lasers, the computers, and all of the other kind of semiconductor technology, really to catch up with that hypothesis or that idea of being able to measure and video a polymerase in real time. Kind of forward wind a decade or more, they brought their first products to the market, the RS2 platforms, great technology, but a relatively limited number of DNA sequences that could be analyzed at any one point in time. About 150,000 positions on this kind of experimental microscope, for want of a better description, could be looked at any one point in time, which was scientifically interesting, but maybe not the scale that's required to match other sequencing technologies. And then really through the course of the last decade, that technology, as technology tends to do, has been incrementally improving step by step with various iterations on the platform that have allowed the number of simultaneous movies to go from 150,000 to 1 million to 8 million. And the company has progressed through there with increasingly successful commercial products, but in a background of a really fast-moving genomics landscape of other sequencing technologies. And in the last 2 years, I think we've reached an inflection point now where the promise of that technology was starting to become a reality, in that we had a combination of really long, accurate sequencing reads, often 15,000 or 20,000 letters long, with very high precision at each letter. And the price points and throughputs were starting to be much more competitive versus the other sequencing methodologies that are on the market today that have been the predominant way that people have been doing large-scale genomics. So we were fortunate enough almost 2 years ago now to raise some additional funding in the stock market. We timed that particularly well, maybe fortuitously, but re-energize the company, reinvest in R&D, reinvest in a commercial team, such that we're at the point now where that investment is starting to bear fruit with brand new products that I think are going to be transformative in the way that people look at genomic sequences as a whole. So we are kind of, give a bit of background on the company as a whole. So we are now about just under 800 people globally. We're headquartered out of Menlo Park in the Bay Area of California, actually right next to Facebook, if anyone's familiar with their massive campus that they have over there. And we've got increasingly a number of sites globally, including a site in San Diego and Baltimore in the US. And I came on board middle of last year to set up the commercial team here in Europe. And we've now got a physical infrastructure, an office and a lab space in London, just near King's Cross as well.
So are you able to talk about both of the new products, Onso and Revio?
Absolutely, yeah. So,
sure.
So Onso, we'll come back to in a moment. I think Revio is one that follows that timeline and trajectory that I was just alluding to in the last comments. So Revio is our brand new high throughput long read sequencing platform. And it really is a step change for us in the capabilities of our long read sequencing. Up until now, the most recent platform we've been selling is one called the Sequel IIe. And that takes one of these kind of small semiconductor chips that has 8 million wells on it and sequences those. What we do on the new platform is now 15 times higher throughput, almost a quarter of the cost of where we were previously on generating a genome. And on the new platform, we can now sequence comfortably over 1,000 genomes per year. We can actually do the equivalent of 4 human genomes of very high depth and precision per day. So huge sequencing power to help people understand rare genetic disease, cancer, biodiversity, all sorts of other areas where genomics is being utilized today. And that platform, we believe, will be really transformative in the next generation sequencing space, because for the first time, being able to generate a sequencing read of a genome with long, accurate reads is comparable of price to where we have been previously with the other short read sequencing platforms on the market. And happy to go on for a long time about why that's important, why the length of the read is important. Maybe some context there will be useful. So Onso, in contrast, is a new short read sequencing platform. And we've certainly had some of our investors, some of our customers ask, well, PacBio has this history in long read sequencing. Why have you also spent money and time and energy making a short read sequencing platform? And I think the logical answer to that is sometimes you can't get nice large fragments of DNA to do your sequencing. There are many biological circumstances where bits of DNA are already naturally fragmented, cut up into small pieces. That could be the DNA at the scene of a crime, for example, that's degraded. That could be DNA that's naturally just floating around as little fragments in your blood. And those in particular are the area that we're interested in, being able to utilize small fragments of DNA, either from a blood sample or from a cancer sample that's been treated in formalin and fixed by your pathologist. The DNA is often very fragmented, and we need to look at those tiny fragments of DNA very precisely. So we now have a portfolio with two sequencing platforms, the Revio, high throughput, low cost, very long, very accurate reads, and the Onso platform, similar high throughputs, but smaller sequencing reads, but uniquely differentiated versus other competing players in that space, in that we have more than an order of magnitude greater sequencing accuracy. And for some applications that's relevant, for some it isn't. But if you think of that application of looking at a blood sample and looking for maybe the one read in, or one fragment of DNA in a million that is mutated in a way, it's come from a tumor that you have in your body somewhere, and a little bit of DNA is broken off and it's floating around your blood. To be able to detect cancer early, you need to be able to accurately say when you sequence that read, it really is a mutation in EGFR or one of the other cancer-causing genes, and not just an error in your sequencing. And so we're excited about how Onso will allow us that additional precision to really find the early signs of cancer, or maybe to follow someone post-operatively during their treatment course to work out whether their cancer truly has gone away. When that clinician says to the patient, "You're clear of cancer," at the moment, that statement probably should always be caveated with, "You're clear of cancer as far as we can detect," and the sensitivity of whatever detection methodology is always, it's never perfect. Being able to take that down and be 10 times more confident when we say that now, I think is going to be transformative in the oncology space. So we're really excited about how that exquisite precision and accuracy of Onso will play out specifically when we're looking at cancer.
Sounds really exciting for the whole field. But are there any other applications for both of these? Obviously, they sound like there'd be many, but
yeah, I mean, let's kind of go back to the long read side of things, the Revio platform, and I'll try and explain in terms that hopefully the audience can understand how long read sequencing is beneficial. So we'll use human genomics as an example, although, as I said earlier, many of our customers and researchers around the world are using our technology in all sorts of plants and animals. Maybe we come back to that later. But in human genomics as a whole, I think most of the general public now recognize that you inherit 3 billion letters of DNA from your mother and 3 billion letters of DNA from your father. Those intermix and end up as your genome, your unique book of life that is your genetic code. In any one person's genome, there are probably about 4 million what are known as single nucleotide variants, little spelling changes where maybe an A is a T in your genome, it was an A in someone else's genome, or a G is a C, some other variation like that. So about 4 million of those scattered across the 3 billion letters of our genome. There's roughly another 1 million, what are referred to as indels, insertions or deletions, typically less than 50 letters long, that maybe you've got an extra few letters in that bit of your genetic code, or maybe a little bit has been deleted. So in average, you've got about 5 million locations that change within that 3 billion letters as a whole. Most short read sequencing technologies today cut the DNA up to be able to sequence it into tiny fragments. So it's a bit like taking your book of life as a jigsaw and smashing it up into 300 letters long. Maybe in those 300 letters, if you wanted the kind of analogy, it's like you take your whole book of life and you break it up into words. So I've got a the. Where does that go in the book of life? You frankly no way of knowing. In that 3 billion letters long, if you've only got 5 million variations, more than half of the words that you're sequencing with short read sequencer have no variation in them at all. You basically have to guess as to where they go. Whereas our long read sequencing, we take the 3 billion letters and cut it up into roughly 200,000 pieces long. 200,000 pieces, about 15,000 to 20,000 pieces long. Each of those is more like a paragraph. And within that paragraph, there are multiple different locations, often on average about 25 locations in that paragraph that are, distinguishing for whether it came from your mother or your father. And that allows us really to assemble the book of life more effectively and really definitively say this sequence came from your mother, or this part came from your father. And that layer of information, I think, is going to be absolutely essential to really understand why your genetic code predisposes you to some diseases or others, why it makes some of us have brown eyes or blue eyes, or whatever character or feature you want to ascribe to your genomics. So that in itself, that ability to uniquely say where the DNA has come from, is really the power of long read sequencing. And it's allowing all sorts of new insights and discoveries that just couldn't happen when you cut it up into tiny pieces as the prior technologies have been. Now, historically, to do that had been 4, 5, 10 times more expensive than to cut it up into the smaller pieces. The economies of scale on short read sequencing had been significantly more cost effective. And really the revolution with Revio is that we can now generate these long, accurate sequencing reads at a price point very, very similar to what we can do with short read sequencing. So today now on the Revio platform, we can now produce a high quality human genome at very exquisite accuracy for under $1,000. So a similar price point and benchmark to where the short read sequencing companies have been in the last few years. And I think that's going to now encourage many researchers to transition over from this sort of low fidelity genome that they've been using on short read sequencing to a hi-fi genome that PacBio can produce.
And so who is it aimed at?
Really anyone doing academic research in genomics. I think that's our main audience at this moment in time. It really is a research tool. We think over time it will transition more into the clinical environment. I think brand new instrument, we only announced it last week at the American Society of Human Genetics meeting, but there's a lot of excitement. I think there is some obvious areas where we're already seeing excitement, and one of those is rare genetic disease. If you think about it, well, the term rare genetic disease does suggest it's maybe not that important. There are thousands of different genetic diseases, different genes in an individual's genome that can have deleterious variations in them that lead to all sorts of different diseases. And so cumulatively, while they're individually rare, they're collectively fairly common. As of today, fortunately here in the UK, the NHS has adopted next generation sequencing, the short read technologies, to try and help families that have an affected child with some significant success, certainly improvements on where the previous standard of care had been. But still more than half of the individuals that have short read genomes sequenced with that technology today don't get a diagnosis. They aren't able, because of the resolution of that platform, to determine what the underlying genetic variations are, the spelling mistakes in that child's genome that are causing disease. And we believe that our long read sequencing, because we can really work out where those paragraphs of the human genome have come from rather than the words, we believe that we'll be able to significantly improve that diagnosis rate over time to help many more of the families find an underlying cause of their condition, their disease. So I think that's the most obvious area where we're already having a lot of people excited, but it extends all the way through disease as a whole. Almost every disease has a genetic component, whether that is rare diseases that we were just touching on, whether it's cancer, which is essentially malfunctioning genetics that's gone a bit crazy and your cells have grown up in an uncontrolled fashion, understanding how those mutations arise and how they escape your immune system, how they respond to drugs. The genetics is an integral part of that. So the cancer space as a whole will be one where I think there'll be a lot of utility. But it goes all the way through, as we've seen, in the last few years in the pandemic, understanding the variations in a virus like SARS-CoV-2 and how that spreads between individuals can give an understanding of the pathogenicity of a virus or the ability of that virus to spread to evade vaccines, for example. So it really can touch all aspects of healthcare and then all aspects of biology. Anyone that's in the agricultural sector trying to grow plants or breed animals at this moment in time, if we want to feed the world of almost 8 billion people that we have today, let alone kind of future growing populations, we're going to need to better understand the genetics and maybe be able to grow plants that are tolerant to drought, or maybe tolerant to flood if the last 24 hours rain is anything to go by. And understanding the genetics of how some species can do that very effectively versus kind of the current agriculturally used crops like corn or wheat or barley, how they are susceptible to those types of conditions is, again, a function of their genetics. So really the span of where genetics can be used is vast, and it does make it a hugely exciting area to work in. There's many of the team that just love the scientific conversations and the global impact that this technology can have.
You were mentioning about things like drought tolerant plants. Is that something where you could theoretically take something from a drought tolerant plant and apply it to a plant that isn't drought tolerant?
Theoretically, yes. And I think the genetic engineering tools are continuing to advance significantly to allow that to be done in a safe way. Now, clearly, safety is a really important part of anything that we're doing with species and the environment as a whole. And, different countries and jurisdictions around the world have their right safety policies. First and foremost, actually understanding the genetics of that species accurately in the first place, so you know how each of those letters in that plant's genetic code allow it to respond to the environment is critical. And we're playing an integral part of that, making these sort of reference genomes, the first book of a wheat species or a corn or a maize. But we're also seeing our technology being used in controlled environments in laboratory situations, such that if someone is experimenting on changing a bit of that plant's genome to try and make it more drought tolerant, that it is precisely that specific bit of DNA that's being changed and not accidentally some other region of the genome that could cause other impacts that are unknown. So I think in both aspects, the underlying research, but also the quality control and safety control of that, the genomic tools that we produce are playing a really important role.
And the platforms that you have just launched, are those things that are constantly being refined, or is that you're just done now?
No, like most companies, we want to continue striving and moving forward with the technology. So it's a constant iteration, while our R&D team are really proud to be able to now tell the world about what they've been working on for the last couple of years and share the excitement of where these higher throughput, lower cost instruments are going to allow genomics to go. They're sadly now back in the lab working on what the next iterations are going to be. How do we make this easier to use for people in the lab? How do we make it cheaper again? How do we make it even higher throughputs? How do we make it more accurate? Lots of different vectors where the technology can work from. And we have an amazing R&D team that have some of the world's experts in all sorts of areas. This genomics technology is really an apex of all sorts of interesting areas of science as a whole, everything from the very latest machine learning. We actually have a very active collaboration with Google. So we've partnered with Google on some software development using their expertise in machine learning to get the very most out of the data of our instruments. We've got people working on semiconductor manufacturing, really at the cutting edge of making tiny holes in little bits of a chip. We've got engineers working at lasers and looking at photonics and the physics associated with how light bounces around. So a huge group of super smart people that come together to try and bring all of their areas of expertise into a single product that is used in this genetic space, but is a culmination of all sorts of interesting bits of science.
Is it something that you have to teach people how to use, or is it easy for the researchers that will be using it?
I think we normally do teach any of our new customers how to use our equipment. I think there's room for us to continue to get more and more effective at having a product that's just easy to use straight out of the box. And certainly there's an active part of our R&D that's doing just that. But normally when we sell one of our instruments to a specialist research lab, we'll go in, we've got our own application specialists, our own engineers that will go to that customer site, to that academic institution or that researcher, and we'll show them the ins and outs, the bells and whistles of the platform. We'll walk them through the lab with their pipettes and show them how to go from their biological sample to get the best quality DNA out of that, to manipulate that, to do the sequencing on our platform, to show them how to do the analysis as well, and the very latest bioinformatics tools that are used for analyzing the data. it's a complex process from that biological specimen to the scientific insight that can come from it. And so we tailor that training really for each of our different customers that we sell our instrumentation and reagents to.
And I assume it's available globally.
It is available globally. We have announced the Revio and the Onzo platforms just last week at the American Society of Human Genetics. We are actively taking orders now for the Revio platform. We're actually still doing some customer evaluation of the Onzo platform. We'll start taking commercial orders for that probably at the beginning of next year. We just want to finalize some performance in customer hands of that. And then we expect to be able to ship the first of the Revio platforms in the first quarter of 2023, the first Onzo platforms probably in the second quarter of 2023 next year. We wanted to announce it now, give people time to acquire the relevant academic funding, to give them an advanced heads up as they're planning their science for next year. And I think a lot of the reception that we've had has been very positive to giving that little bit of advance notice of the trajectory of travel of our technology.
Excellent. It's a really good overview. Is there anything else that we didn't cover that you wanted to cover?
We've talked a lot about DNA today. So one other important announcement that we made at ASHG is some innovations around looking at the genes that are expressed out of a genome. We have a kit that we call MassSeq. Don't need to worry too much about the naming. It's a collaboration that we've done with a US institution called the Broad Institute. They're an amazing kind of factory setting for genomics as a whole. They've done a huge amount of very large-scale genetics projects on DNA, but they've got some great expertise of looking at RNA, looking at how the genes are expressed in a genome. And again, the really kind of transformative science in the last decade on understanding how genes are expressed, how much of any one gene is expressed in any one cell or tissue, has really been driven by short-read sequencing. That was a transition from previous technologies of little microarrays, sort of DNA spotted on glass slides. They've transitioned from that to actually sequencing little fragments of the genes. But the problem with all of those studies in the last decade is that they've been limited in the same way as DNA sequencing has been. The genes are often in a human genome, 1,500 letters long on average. There's some variation from one species to another. Sometimes they can be really long. But again, if you're cutting those up into tiny pieces, you can't really work out exactly the full length of that sentence. You get a word of the sentence, and maybe you can count those and get a rough feel for how much of that gene is expressed in any one cell or tissue. But you don't get the full biological context. You don't get the sentence, the full transcript at any one point in time. And again, that's where our longer sequencing technology has a distinct advantage. So we've been working with the Broad Institute to generate a new protocol that can actually sequence the full length of that gene, allow you to understand how much of it there is in any one cell type, but also exactly what that sequence of letters is in that gene. And what we're starting to see from that is in one cell type to another, it's not just the amount of a gene that changes, it's how it stitches together its words. And sometimes the exons that gene is made up of, it will skip one or more of the words in the sentence. You'll end up with a transcript that's different. And then that transcript will generate a protein, and that protein, because it's missing a chunk that was in the other kind of format or that other isoform, behaves completely differently. There are situations where that differential splicing of exons in one situation can mean a gene is protective of cancer. In another situation, it can be exactly the opposite. So understanding that biological context, we believe is going to be critical to really, again, just getting this new level, a new layer of biological insights into what's going on. And the kit that we have just launched again at ASHG will fit on either of our long-read sequencing platforms, our current Sequel IIe instrumentation, but also the new Revio platform. And it allows people now to do that full-length transcript sequencing at a scale that's now comparable in price point to just sequencing little fragments of RNA that is possible on other short-read sequencing platforms. So I think we'll see a significant transition. People looking to analyze expression in single cells and work out how they're all behaving and interacting with one another will get this new layer of biology. And that's really what we're trying to do with all of our technology. We feel that we produce the world's most advanced sequencing technology, But it's really the exciting stuff that our customers and the researchers in this whole community do with that is the magic. We make picks and shovels and other people find the gold. So we're delighted to see how some customers have started to use that already. And over the next few years, I think we're just going to see an explosion of really interesting science in all sorts of plants and animal species as a result of understanding how our genome is in the first place with our DNA sequencing, and then how it actually manifests and interacts with the environment, and how our genes are expressed with that RNA sequencing technology, that mass isoseq methodology that gives us the full picture there as well. So lots of exciting stuff to come. We haven't even touched on epigenetics. There's another layer of control that goes between DNA and RNA, little chemical marks on one's DNA that can be a signature of the exposure that our DNA has had to the environment. And we can also natively sequence that now as well. So when we do DNA sequencing, all previous short read sequencing technologies to date generated A, T, C, and G fantastically accurately, but with some of the caveats and challenges of maybe not being able to map those unique 300 base pair fragments very well. What we do on our DNA sequencing now is we sequence A, T, C, and G very, very accurately, these long stretches of DNA from 15 to 20 kb. But we're also, because we're doing this movie of a polymerase in real time as it generates that complementary strand of DNA, we're able to look at not just the base that's being incorporated, but how long the polymerase takes to incorporate that base. And that kinetic signature, watching the movie, watching the polymerase in real time, gives us little glimpses of other modifications to the DNA, other epigenetic marks. And one of the most common in human or mammalian genomics is known as methylation, the little chemical mark on the cytosine base. So your little C base, when it's in a context of having an adjacent G base, or a CpG context, as is referred to, can often be methylated. And in some cases, it's methylated, in some cases it isn't. And that can tell us something about the control of expression of genes. So again, a whole different layer of biology. And we generate that for free. If you're doing DNA sequencing with unamplified material from a human blood sample, you now get all your A's, T's, C's, and G's, but also all of the locations, the 30 million roughly locations in your human genome that are methylated at that point in time. And that gives you a sort of interesting hint as to how the environment is changing your DNA. And we see it changes over time. If you were to take a blood sample and blindly sequence that with our technology, you can determine pretty accurately the age of that individual it's come from, irrespective of knowing nothing else about that individual. Because as we age, as we go through life and are exposed to our environment, and maybe we eat too much or drink too much, or maybe we smoke or whatever, each of these things can damage our genetic material in some way. And some of that manifests as these little epigenetic changes. So you can get a reasonably accurate estimate of someone's current kind of life exposure of their DNA by looking at their epigenetics. Epigenetics controls why one cell is a hair follicle versus an eye cell or a bit of tooth. That's completely controlled by different epigenetic characteristics. Our DNA doesn't change, but the little methylation patterns and how it's tightly bound into a 3D structure will change how one cell expresses genes and another doesn't. So again, a whole layer of biology that we can now detect very accurately, high throughput, low price points, that will just, I think, result in an explosion of new discoveries over the coming months and years.
Our second longer interview this week is all about space, something that I've been interested in since I was young, and no, that wasn't before Yuri Gagarin actually went up into space. The subject here is fascinating, and it is on developing the first stem cell research institute in space. Sierra Space and UCSD, or the University of California, San Diego, have formed a new agreement with the goal of defining the future of human healthcare research in space. So I had lots of questions, and for a change, my son was actually impressed by the subject and actually asked me about it. So here's the interesting conversation with Marc Giulianotti, Senior Manager in Space Biomanufacturing at Sierra Space.
All right. So I guess to kick things off, we'll go with the easy question first. If you could give me a bit of background on the history of Sierra Space.
this is a fun topic to talk about, Jim, and I really appreciate the opportunity to come on here and have this conversation. I think it's a lot of fun, and it's also really exciting times that we're living in. Yourself covering the biotech industry, that there's been a lot of changes in technology lately that are really exciting and seem to be on the precipice of personalized medicine, improvements in, regenerative medicine, and all the different spaces. I mean, it's just exciting to see that explosion of technology in that intersection. And Sierra Space is really, we're right there as well. We're founded with the understanding that we're right there at the next industrial revolution. So while we're seeing this in biotech, we see this next revolution happening in low Earth orbit. And this is going to be built on the 60 years plus of government investment in the space-based industry, and exploration. And now we're really transitioning to these commercial opportunities that low Earth orbit can provide. As a scientist, I'm hoping that I'll get your listeners excited about this opportunity. I think sometimes when we're at terrestrial labs, we're accustomed to the technologies that we have, but we're going to have this opportunity to use this exciting platform in space. And it's really, we're right there. We're coining it as the orbital age. So you're in the biotech world, and we're hoping that the biotech world really embraces this orbital age. So, at Sierra Space, we're a couple years old now, raised over $1.5 billion to move forward on this. But it's, we have over 1,000 employees, and though we're a year and a half plus old, we're really built on the legacy of Sierra Nevada Corp, which has 30 years of space-based expertise, including over 500 missions. So we're the independent division originally out of that, and now we're an independent company, Sierra Space, really looking to capitalize that, become the end-to-end technology platform provider for all our customers in this LEO economy. So this includes getting to space, to low Earth orbit, is what we call it. So the place where we can put a satellite and have it in continual free fall, so you get this microgravity environment. So we've got the capabilities with our space plane, which we call a Dream Chaser. And in my world, I love the Dream Chaser, which will start being operational in 2023. But the Dream Chaser is a plane that can actually touch down here on Earth right now. The capabilities are all about capsules. So when we return cargo or people from the International Space Station right now, they sit in a capsule that has to splash down in the ocean or in the middle of the desert. You can imagine if we're making some new cell therapies in space, we don't want those crashing down in the middle of the ocean and then bobbing around for a couple of days. We want to be able to potentially deliver them to the point of care or deliver them to the next stage manufacturing facility. So that's one of the things that we're working on. But we have capabilities that will enable us to have habitats in space. We'll have research and development facilities in space, have full-scale manufacturing facilities in space. Lots of exciting things. We're really, really looking to be driving this orbital age that we're coming to.
You mentioned all of those things. Will that involve people being up there, or will it all be done remotely?
Yeah. So right now, the opportunities to go to space are pretty limited, right? So, if we go back even 10 years ago, unless you were, NASA or another government astronaut, it was very unlikely that you're going to go to space. So NASA has opened up the opportunity to have commercial crew go to the ISS. So we're just starting there. But at Sierra Space, absolutely, this is part of our vision is that it's not going to be your typical NASA astronauts anymore. We really envision, and this is part of, I think what you're bringing on me to talk to a lot about today is collaboration with UCSD. We really see the scientists actually going up there and doing the research. When we get to the point of full-scale manufacturing, we'll obviously have people up there maintaining the facilities for manufacturing, but the researchers can go up there. I mean, everybody talks about the tourists, and that'll definitely be part of it, but we see the economy being much larger than that tourism.
Like the finances of it, is it something that you was talking about inspiring people? Is it something that is beyond the reach of companies, or do you think that is something that's potentially going to happen?
Yeah, no, I think that it is. And this is the vision. So I think what is exciting is that we've seen this adoption of commercial players in this economy. So, everybody knows SpaceX, and really what they've been able to do is provide services, have capabilities that are reusable to drive down the costs. That's a big part of our business plan as well. As I mentioned before, we have the legacy of what we were founded on. We have I originally came from working at the ISS National Lab, so the current space station. So we know where the key drivers to drive down costs are and efficiencies are. And that's the goal. I'm currently working on programs and working with UCSD to understand where the true return on investment will come for researchers. So yeah, absolutely. We wouldn't do this if there wasn't a clear vision that the costs will be in line with what the benefits are going to be.
Hopefully that means at some point then I get to go up and interview people actually in space. That would be awesome.
Yeah, that would be cool. I think we're going to see uses that range from, a couple days to people living and working there for months at a time. So I think you're going to see this full economy.
So you mentioned UCSD or San Diego. Could you tell me a bit about the collaboration with UCSD, how that came about and what the partnership is?
Yeah, well, I'd like to say it started when I did my undergrad there so many years ago, but that's not how it happened. It's definitely been a partnership with some of our key leadership, and I can touch on that. But UCSD, not to brag on my alma mater, but they've always been at the forefront of pushing research and development, really driving innovation. Definitely in the regenerative medicine and biotech space, San Diego's a biotech hub. So they have always been looking to see how they can expand the capabilities available to them. So they started working on the ISS. They did a lot of work primarily focused on stem cell research, organoids, and early stage cancer development, really trying to understand how when you put your research into a platform that's orbiting the Earth, so has this microgravity variable to play with, how that has an effect positively or negatively on some of these biological systems. So, here on the Earth, we understand that we can change the temperature easily. In the lab, we can change the pressure, the gas contents. The one thing we can't do readily on any sort of scale is remove gravity. And so, as a researcher, if somebody told me, hey, I can walk into this lab and I can, manipulate gravity, I would definitely take advantage of that. It's just not a possibility here. So with the current ISS, it is a possibility, and it's really amazing. I mean, we as humans and biology, we have become adapted to living in a 1G environment. And when you adjust that, you see a lot of different transformations that, the groups at UCSD are already starting to see how that can apply for their fundamental research, but also potentially for therapeutic value. That's how it originally started, and we've been working to build with them to go from, what the capabilities are on the ISS to what they would want if they could design to suit their needs now. That's where we're at. The ISS wasn't necessarily built to be a research and development facility or manufacturing facility. Definitely not, for biotech. But now we can. And so that's where we're at with the UCSD team. There's a lot of work being led by Kat Jameson, who's the director of the Sanford Stem Cell Institute. She's also got the Integrated Space Stem Cell Orbital Research team that she's leading. And Denny Sanford is heavily invested with this effort, through philanthropy. And our CEO, Tom Weiss, and the rest of the leadership team is really pushing us to develop this ecosystem with this partnership.
What's the relevance and the benefits of actually doing research in space?
Yeah, so I mentioned that, the real critical thing here is in my mind, is that you can eliminate gravity on a large scale. If people understand or have seen what we've been doing in microgravity, they know that eliminating that gravitational force has effects. So the classic examples is the biotech industry has been utilizing microgravity to make more perfect crystals for structure biology determination. And the reason is because as those proteins come together and form these crystals on Earth, you get convection forces from the gravity, and it disrupts this uniform perfect crystal formation. We also know that when humans go to space, they get accelerated disease-like symptoms. They get accelerated muscle loss because they're not exposed to the forces of gravity. Their bone turnover is different. Their immune function is different. And then when we take it to a cellular level, we have seen that stem cells behave differently. So if you're trying to create a therapeutic product from a stem cell, some of the challenges are, either differentiating it down a specific pathway or maturing it down a specific pathway and being able to manipulate the gravitational force in this way we're seeing has a profound effect. So it ranges from materials, which we might not get into today, but that's along the same lines of the perfect protein crystal. You can imagine that with other inorganic type materials as well, to human disease relevance, and then also to the biological manipulation. So even imagine creating organoids, tissues, different scaffolding, because now you don't have to worry about cross-linking of your scaffolds because you've eliminated the gravitational force. So there's lots of applications. And now the task is to put in place the technologies that will enable us to drive some of those innovations in the return.
What kind of research will you be doing in space, or what's the first kind of steps?
The UCSD team, for sure, the goals there are to really expand on their stem cell, organoid, and cancer research programs. That's where their main emphasis is right now. But they have a really strong materials group there, and so we're looking to expand into that. The good thing with the biologics is that they can be really low volume. So the return products, you can imagine that a cell therapy product might be in a 50 ml vial, right? But there are all the regulatory approvals, and we'll have to work through how you actually manufacture in space. And that's part of what we're working with the UCSD and some others on, is how we can have these systems where we can actually get ISO 9001, move towards clinical therapeutic use. And so that will definitely be working in those areas. But there's novel materials like semiconductors, that we can potentially make in space, where we're at the point now where we're getting so small and so thin with those that gravity is really starting to play an impact on the manufacturing capability, and to envision, manufacturing materials at scale where the gravitational force is influencing the quality of the products is some of the key areas that we'll be going after.
Sure. Yeah, you mentioned regulation. That'll be an interesting one. Any ideas yet as to jurisdictions for space or?
So one of the cool things, if there are younger people, I don't know what your audience demographic is, but if I had a kid that was interested in law, but also had a space bent to them, I think this is a super exciting area. Space law is going to be the thing that governs this orbital age. I mean, there are a lot of unknowns, but there's a lot that we can rely on current laws and regulations. But yeah, I think that's definitely one of the things that we provide as a commercial space station. That's a differentiator from having a government use facility, right? So as a commercial space station, we have the ability to work out the legal implications, develop contracts that provides the capabilities to secure IP for our customers, for example, and work to custom build if manufacturing regulations are required.
Could you tell me a bit about the orbital reef?
Sure. So, the orbital reef is primarily a partnership with Blue Origin. So as I mentioned to you before, NASA's moving away from wanting to own and manage everything themselves. So they've seen a lot of success. Currently, the way they get their cargo, their research experiments, as well as the resupplies to the astronauts that are on the space station, is they work with commercial partners now to transport that. And we actually have one of those contracts. So we'll start flying the Dream Chaser to the space station to resupply cargo starting in 2023. So there's this transition where NASA doesn't want to make and manage all of these facilities themselves. They want to contract that out, or they want to have relationships with commercial organizations. So the International Space Station is currently set to decommission around 2030. And so again, NASA's vision for the future, they want to have a presence in LEO. They want to continue to have a presence there, but they don't want to build and operate it and manage it all themselves. So they're looking to the commercial sector to provide that capability. And so really that's one of the primary goals of the orbital reef, is to meet that need for NASA. So we are working with NASA on that right now. So we currently have some preliminary designs that we're working through with them, with the idea that we'll have capabilities available that will enable them to transition off of use of the ISS before the ISS is decommissioned. But the goal is that this is more than just a NASA facility. This is this large commercial mixed use facility where we're really providing end-to-end capabilities. So from transportation up, full infrastructure there, transportation down, where commercial companies or academic institutes like UCSD will have a place to do their research or their manufacturing. So, from the Sierra perspective, we'll be involved in obviously the transportation part with the Dream Chaser. We also have our inflatable habitat, what we call Life. And so one of the challenges of building a space station is getting all the parts up there. And so what the Life habitat is, the big advantage to it is it's an inflatable. So what it enables you to do is to fly a volume that would typically maybe take you 4 launches to get that volume of space up into space. We can do it in a single launch. And so again, the goal here for commercial use is to continue to drive these costs down. And so by obviously limiting the number of launches to have a serviceable platform is key to that. So it's exciting. We're moving towards working every day. I have calls on my calendar every day with the Blue Origin team on our orbital reef plans, and we're hoping to have capabilities up there in the 2027 timeframe.
Is it going to bring down the cost of drugs? Is it going to speed up getting drugs to market?
Yeah, I'll talk on 2 examples they're out there. There's some publications on them, but I'll start with work that Merck is doing. They have a PD-1 inhibitor, their Keytruda. So it's $20 billion a year in sales. So it's a monoclonal antibody. And so when they manufacture that, the formulation that they get is a dispersed collection of crystalline suspension. So it's non-uniform. So from a manufacturing perspective, that's challenging to have a reproducible process that's non-uniform. They've also noticed with that non-uniformity, it's not as stable for storage conditions. And so they've published all of this data. And also the limitation is that you are not able to have a formulation that allows you to have like an injectable or an oral. So the only way to get the treatment, which is very effective, is to go into the clinic for an IV treatment, which, from a patient's perspective is obviously not the best. We would all, rather take a pill or at a minimal be able to take an injectable home and do that yourself. The limit is this manufacturing of the final product. What they found is they were able to go to space and develop a uniform microcrystalline suspension, which actually, in all their in vivo and in vitro models is just as effective as their other therapeutic, but it has tremendously better shelf life, and they can store it at much more ambient conditions. And they believe that they could develop it into a therapeutic product that would be able to be either injected or orally suspended. So that's $1 billion industry that could be transformed by a manufacturing process that's done in space. So there's lots of other monoclonal antibodies. There's lots of biological therapeutics that are coming on board. And, there's opportunities in that area. We also have the stem cell opportunities where, patients have been receiving cancer therapeutics, stem cell treatments for many years already. But the relapse after those treatments, it can be pretty quickly. And so you can imagine if we could develop a therapeutic product that is more potent, more efficacious, and extend the patient's life, prevent them from having to come back for a second therapeutic treatment. Not only are you improving the well-being of the patient, but you're driving the costs down because now you're not having to come in for second treatments. So, those are just a couple case studies, but we're just at the beginning. So I can envision people a lot smarter than me will figure out lots of other ways to utilize these new facilities.
timelines and milestones? What kind of timelines are you looking at for this project?
Yeah, so we're working right now. So as I mentioned, we've got the work going on with the Orbital Reef program, and that's UCSD is heavily involved with that. The goal is that we'll have something on that platform operational in 2027. But obviously, you don't just get to 2027 and turn it on. So there's a lot of planning that gets into there. So as we're developing the infrastructure and the capabilities to support that work, we're still utilizing the ISS right now. So we're partnering with the ISS. So NASA and the company that manages the U.S. portion of the ISS, CASIS, are very supportive of this work and helping us to do a lot of experiments on the ISS to, really test some of these hypotheses that the teams have. So we're starting now, and, we're driving towards milestones on all different fronts with the goal so that hopefully in 2027, you can look to the skies and know that our orbital reef is up there already.
Not holding you to this, but looking into a crystal ball, what do you see as the future for research like this in space?
You touched on it a little bit earlier, saying that when we talked about just the common person maybe going to space, I really think that there's going to be that inflection point where it's going to be common for us to have, less than 6 degrees of separation from someone that went to space. So there'll be that aspect, and that will be because we will have products that we're using here on Earth that have their legacy from use on these platforms. So it may not be that there's on the label that, hey, this was made in space, but it'll either be some research and development that was done that has enabled that product, or that product may have actually had a component or even the whole product manufactured in space. And that will just become commonplace. So I won't tell you that I think we're going to have livers that you can go in and check in with on a Friday night, or you can replace your heart. I won't let you pin me down to the exact technology, but I think that that's what's going to be commonplace. I think that's the exciting part is as a business, we see the clear markets we're going after now. In 10 years from now, it'll be exciting to see what these new markets are that generate because of the opportunity.
Now we stay in the US for our weekly contribution from JLL and Travis McCready. Hey Travis, what's on the radar this week?
Hi Jim. It was a big week in the US as we all labored to digest and understand the results from our midterm political elections. One takeaway that was clear from voter feedback: the age-old adage about what inflames voter passions continues to ring true. It's the economy. If the life sciences were a voting bloc, indeed, that saying would certainly apply as well. It has been many years since the industry labored through such a volatile, nerve-testing macroeconomic environment. Costs are going up, supply chain is constrained. With a sallow IPO market, exit strategies need to be reevaluated, funding mechanisms are showing hairline fissures, and even the consumer confidence, as it were, of scientists and technicians are being tested. However, as I always counsel, science and scientific progress is a process and a journey, not a destination. And as such, the angle of declination of the industry's growth sometimes belies the extraordinary heights that the industry will achieve over time. Nevertheless, in challenging economic moments, it is a good exercise to return to fundamentals. For investors, in particular, seeking to understand where the puck is going, to use a hockey phrase, we look for environments producing high concentrations of quality early-stage companies. Those that meet an unmet medical need, have an innovative, proprietary, and differentiated technology, occupy significant defensible market position with favorable pricing, and the like. It is well understood that many of the technologies and teams that meet these criteria emerge from academia or research institutions and are federally funded. Not surprisingly, life sciences investors tend to align behind the breadcrumbs and economic impacts left by agencies that invest heavily and successfully in life sciences innovations. In the U.S., the funding agencies that commonly come to mind are the National Institutes of Health or Biomedical Advanced Research and Development Authority, BARDA, the National Science Foundation, the National Aeronautics and Space Administration. Wait a second, NASA? NASA indeed. Long known and appreciated facts about this spacefaring agency, in addition to spurring growth in extraterrestrial activities like space travel, aviation, and space mining, out of necessity, NASA has long invested in scientific innovation that help us understand the human condition. For decades, NASA has been pumping out unique, differentiated research in cell biology and physiology, muscle physiology, radiation biology, and neuroscience that have translated and integrated into commercial life sciences applications. The list is too long to count, but think LASIK eye surgery, medical sutures, medical light therapy, organ-on-a-chip technology, biometric sensors, and high-resolution imaging, just to name a few. Not to mention the countless ongoing cell culture, tissue, and small organism experiments being performed in microgravity aboard the International Space Station, providing the scientific community invaluable understanding of biological mechanisms and processes. A few weeks ago, NASA produced its economic impact report for the year, demonstrating $71.2 billion of impact for fiscal year 2021, supporting nearly 340,000 employees and $7.7 billion in tax revenue generated. Importantly, NASA's activities touched all 50 U.S. states, albeit not evenly. The report available online shows where the activity is concentrated geographically, and NASA's tech transfer website allows you to freely search the innovations and patents that are available for commercial application. Combined, the resources offer a lens into NASA's activity that suggests the agency has much more day-to-day significance to our innovation economy than perhaps it receives credit. Looking forward, as one of NASA's Science Mission Division leaders shared with us at a recent life sciences retreat in Houston, the challenge of long-distance space travel to Mars and beyond is not just about physics and aeronautics, it's also about physiology. In space, we must wrestle with bone density loss, nutrition, radiation and cell damage, and countless other afflictions which, incidentally, have direct nexus to some of the thorniest terran challenges we face, like cancer and osteoarthritis. To that end, NASA is investing heavily in research in these areas, and given the agency's excellent scientific track record, it's simply a matter of time before novel approaches, interventions, and technologies will be available for commercialization that will help the life sciences sector continue to grow. In other words, during this recalibration of the markets, if life sciences investors are looking for indicia of promising future activity, either by way of intellectual property generation or vibrant innovation clusters, I bet that NASA and its progeny are a good North Star. Ugh. Excuse the dad joke. Thanks, Jim, and see you next week.
Great, thanks so much, Travis. Yet more food for thought on space and the future of research. And nice to tie in with the previous interview. Hopefully we will catch up with you again next week as we edge closer to Thanksgiving in the US. Travis McCready is the leader of JLL's Life Science Markets Advisory practice in the Americas, working closely with the global and scaling life sciences companies, developers and investors to achieve breakthroughs. He has more than 25 years of experience spearheading successful ventures related to technology and innovation, including as president and CEO of a $1.6 billion life sciences funding agency. And that's it for another week. I'm going to try and convince my son he needs to work in space, and next week I won't be on the road, except for groceries, that is. Looks like another busy week ahead, planning for another event, BioFit in Strasbourg, and I'll also be getting ready to moderate a webinar later in the month. And we also have a special newsletter coming up next week on children's conditions and diseases. And there's all those videos to do, so I'd better get to it. I hope you enjoyed this week's podcast with a couple of cutting-edge topics, and that wherever in the world you may be, that you have a great week ahead, and you'll join us next time for another Beyond Biotech.